For animal cells to function properly, it is essential that the fluid surrounding them has the same water potential as the cell's contents, to prevent harmful water movement. If water moves into or out of animal cells, it can cause them to swell or even burst. To avoid this, surgeons use saline solutions with matching water potential during medical procedures to prevent cell damage. Similarly, during physical activity, isotonic sports drinks, which have the same water potential as the body's fluids, help replenish lost water and salts while providing energy, without causing osmotic disturbances. Dehydration can occur when fluid loss from sweating isn't replaced by adequate water intake, leading to a decrease in blood plasma water potential, which negatively impacts cell functions. The diagram on the whiteboard illustrates the movement of water molecules in and out of a cell. Here, the image illustrates the effects of different solutions on red blood cells, highlighting the concept of water potential and osmosis in animals. The whiteboard is divided into 3 sections, each showing red blood cells in a different type of solution. In the left section, labeled isotonic solution, the beaker labeled contains a solution with the same water potential as the red blood cells. The cells retain the normal biconcave shape, with water molecules moving equally in both directions across the cell membrane. The middle section, labeled hypertonic solution, shows a beaker, labeled B, with a solution of higher water potential, less concentrated solutes. Water molecules move into the cells, causing them to swell and potentially burst through hemolysis. In the right section, labeled hypertonic solution, the beaker labeled C contains a solution with lower water potential, more concentrated solutes. Water molecules move out of the cells, causing them to shrink and become crenated, with a spiky appearance. This image effectively demonstrates how osmosis impacts red blood cells in various environments and underscores the need for a balanced water potential for proper cell function. When an animal cell is placed in a hypertonic solution, such as distilled water, which has a higher water potential than the cell's internal environment, water molecules move into the cell, causing it to swell. Since animal cells do not have a cell wall to contain this pressure, the cell eventually bursts due to the internal pressure. This process occurs because the influx of water is greater than the cell's ability to expel it, resulting in cell lysis. A normal cell maintains its original size and shape, as the concentration of water and solutes inside and outside the cell is equal. In this state, water molecules move in and out of the cell at the same rate, maintaining equilibrium. This occurs when the animal cell is placed in an isotonic solution, where the water potential inside the cell matches that of the surrounding environment. When an animal cell is placed in a solution with lower water potential, higher solute concentration compared to its internal environment, it loses water. This causes the water molecules to move out of the cell, leading it to shrink and shrivel. This process, known as crenation, occurs when the cell is in a hypertonic solution. To sum up, let's take a look at this. The image here clearly illustrates the effects of different solutions that are hypertonic, isotonic, and hypertonic, on both animal cells, specifically, the red blood cells and plant cells. It is divided into 3 columns representing each solution type, left for hypertonic, middle for isotonic, and right for hypertonic. The top row shows animal cells, while the bottom row shows plant cells. In a hypertonic solution, red blood cells swell and may burst via lysis, while plant cells become turgid, with water entering and pressing the cell membrane against the wall. In an isotonic solution, red blood cells appear normal with equal movement of water in and out, and plant cells become slightly flaccid. In a hypertonic solution, red blood cells shrink and become crenated as water moves out, and plant cells undergo plasmolysis, with the membrane pulling away from the cell wall due to water loss. This breakdown highlights the role of osmosis in different environments and the importance of water potential in maintaining cell structure and function.